I'm excited to introduce our next speaker, Niran Murthy, professor at the Department of Bioengineering at UC Berkeley Innovative Genomics Institute.
Okay, so good afternoon, everyone. My name is Niran Murthy. I'm a professor at UC Berkeley in the Department of Bioengineering, and I'm also a member of the Innovative Genomics Institute. The title of my talk today is Therapeutic Gene Editing Enabled by New Delivery Vehicles. And before I start, I'd like to thank the organizers for giving me this chance to speak. Cas9-based therapeutics have the potential to revolutionize the treatment of genetic diseases, and this is because of their ability to cut DNA with sequence specificity and have the potential to treat a wide variety of genetic diseases ranging from Duchenne's muscular dystrophy to cystic fibrosis. However, before the tremendous therapeutic potential of Cas9-based therapeutics can be realized, safe and efficient strategies for delivering them in vivo need to be developed. And this is quite a challenge because Cas9 is a fairly large protein. It has a guide RNA associated with it. And if you want to correct gene mutations via homology-directed repair, you also have to be able to deliver a piece of DNA into a cell. So to successfully deliver Cas9-based therapeutics, you have to develop strategies for delivering proteins, RNA, and also DNA sometimes, all simultaneously. And efficient methods for doing this have not yet been developed. However, given the tremendous therapeutic potential of Cas9 therapeutics, there is great interest in being able to do this, and a variety of delivery strategies have already been developed to deliver Cas9 in vivo. These are summarized on this slide over here. In general, Cas9 delivery strategies can be broken up into two methodologies, viral-based methods and non-viral-based methods. Both have their pros and cons, which I'll discuss on the next few slides. So viral-based methods in general are more efficient than non-viral based methods for delivering Cas9 and other gene editing enzymes. Methods, the adeno-associated virus has really won out as the most safe and most efficient way of delivering gene editing enzymes in vivo. And AAV right now is actually much more efficient than any type of non-viral delivery strategy. However, despite its very high efficiency, using viruses to deliver gene editing enzymes has several areas of concern. The first one is that viral gene therapy was really designed for delivering native human proteins into patients. And it wasn't actually designed for delivering bacterial proteins into patients. And the problem is that most gene editing enzymes right now are bacterial proteins. So if you deliver a protein like Cas9 with AAV, there are two potential problems that will happen. The first one is the immune response. Cas9 is a foreign protein, so if you constitutively express it in human cells, in patients, for long periods of time, it is very likely that you will get MHC-mediated immune destruction of your transfected cells, which will essentially prevent all the therapeutic benefits from being accrued. In addition, Cas9 and all of the other gene editing enzymes have a certain level of off-target DNA damage. They're not perfect at just cutting their target site. They will start targeting and cutting other regions of the genome. And therefore, if you express Cas9 for long periods of time, AAV will be expressed inside of a cell for about 5 years. So if you express Cas9 for about 5 years in a single cell, the probability that you start getting off-target DNA damage is extremely high. Finally, a large fraction of the human population already has pre-existing immunity to AAV and would be ineligible for an AAV-based therapeutic. So therefore, there's great interest in developing non-viral strategies for delivering gene editing enzymes. There are three formats in which Cas9 and other gene editing enzymes can be delivered non-virally. Those are shown here. You can deliver Cas9 either as a plasmid DNA, as messenger RNA, or as the ribonucleoprotein complex, which refers to the protein complex to the guide RNA. In general, non-viral methods are less efficient at delivering Cas9 than viral-based methods, except for one caveat, which is in the liver. In the liver, it turns out that non-viral-based gene editing is remarkably efficient, and Intellia just had a very successful clinical trial where they delivered Cas9 messenger RNA in solid lipid nanoparticles and were able to successfully edit the liver of 6 patients. It was actually a real breakthrough in the field of gene editing. But outside of the liver, non-viral methods are in general fairly inefficient, and that's one limitation of non-viral gene editing. And so to address this limitation, our laboratory has developed several delivery strategies for delivering non-viral gene editing enzymes. The first one we developed is a delivery vector called Crispr Gold, shown on this slide here. Crispr Gold is a nanoparticle drug delivery strategy that's composed of a gold nanoparticle core, and to that gold core we have conjugated donor DNA, and this donor DNA is designed to correct DNA mutations via homology-directed DNA repair, or HDR, and then on that DNA gold core we then complex the Cas9 RNP. The Cas9 RNP has fairly high affinity for double-stranded DNA, and so the Cas9 RNP then binds this gold nanoparticle core that has DNA conjugated to it, and then we mix all of this with an endosomal disruptive polymer called PAS DET, and that's Crispr Gold. So Crispr Gold is designed to have all the components needed to do homology-directed DNA repair in one nanoparticle, so any cell that internalizes it will have the Cas9, the guide RNA, and also the donor DNA in the same cell, so that when the Cas9 does the cutting, the donor DNA is also there in high concentrations to do the HDR. I should say that this work was done in collaboration with numerous laboratories, including Jacob Koren's lab, Jennifer Doudna's lab, Irina Conboy's lab, and Kazunori Kataoka's lab. Crispr Gold has numerous potential medical applications. The first one we selected was using it to develop a therapeutic for Duchenne's muscular dystrophy. We selected Duchenne's as an initial testbed for Crispr Gold because of the great unmet medical need with regards to treating this disease. Duchenne's is a disease that affects Caucasian males with a fairly high frequency for a genetic disease, and its frequency is 1 out of 3,500. There's no cure for this disease. Every patient who gets this disease pretty much dies in their 30s or 40s. Duchenne's is caused by a mutation in the dystrophin gene, shown here, and dystrophin is a protein that's involved in maintaining muscle health. And the reason we need dystrophin is that our muscle tissues are undergoing constant amounts of stretching because of their contraction cycles, and the stretching, it turns out, is very damaging to their membranes and very damaging to the cells, and dystrophin protects them from that constant physical trauma. However, in patients with Duchenne's muscular dystrophy, they have a mutation in the dystrophin gene. They don't make functional dystrophin, and their muscle tissue eventually dies off, and that leads to their death. So we investigated if Crispr Gold could correct the dystrophin mutation in using a mouse model, and this mouse model is composed of mice that have a C to T mutation in exon 21 of the dystrophin gene. This generates a stop codon. These mice are called MDX mice, and we investigated if Crispr Gold could correct this mutation by homology-directed DNA repair. So we made Cas9. We made Crispr Gold that had a guide RNA that targets the dystrophin gene, and also a donor DNA that was designed to correct the C to T mutation. We then started a collaboration with Irina Conboy's lab. Her lab injected it into the muscle tissue of MDX mice simultaneously with cardiotoxin, and then 3 weeks later, we sequenced these mice, the muscle tissue of these mice, to look for the correction of the dystrophin gene. The results are shown on the next slide. So the y-axis here is the HDR efficiency. So this refers to the percentage of genomic DNA in the region of the injection where the mutated gene was corrected to the wild type sequence, and the x-axis is the two groups. So you can see that mice treated with Crispr Gold in the vicinity of the injection site, 5.4% of their mutated dystrophin was corrected back to the wild type sequence. And in the control, which is just the Cas9 RNP plus donor DNA, it's at baseline level. So 5% is fairly encouraging for a genetic disease like dystrophins. If you did get 5% in the clinic, the expectation would be that you would see a clinical benefit. We also did immunohistochemistry on these mice to investigate if rating dystrophin expression. So the results are shown here. So in this experiment, we stained these histology slides with a dystrophin antibody. And you can see that control MDX mice, there's no positive staining with the dystrophin antibody. In the wild-type mice, all the way on the right, you can see a lot of dystrophin staining. And in the CRISPR Gold treated mice, you can see that in the vicinity of the injection, you see a very high percentage of dystrophin-positive muscle fibers. So this demonstrates that we are able to regenerate dystrophin protein in the muscle tissue. One of the benefits of using CRISPR Gold is that it delivers the Cas9 protein itself, which should get degraded after the editing has been performed. And CRISPR Gold should therefore have low levels of off-target DNA damage. To determine this, we did deep sequencing on 6 genomic sites that had high homology to our guide RNA. We called them OT01 to OT21, and the results are shown here. So the numbers under the table that says CRISPR Gold, you can see that those refer to the percent mutations. So the highest off-target percent mutation was 0.17%, and everything else is below that. Most of these numbers, if not all the numbers, are sort of at the level of deep sequencing error. So we can confidently say that CRISPR Gold is generating very low levels of off-target DNA damage. Finally, in this study, we also investigated if CRISPR Gold could improve muscle strength in these mice. So for this experiment, we took MDX mice and gave them numerous injections in their various muscles, and then did a 4-limb hanging test 2 weeks after the injections. And this is a test where we take the mice and put them on a barbed wire mesh, and then turn the mesh upside down and see how long it takes for the mice to fall. And what we can see is that in the mice treated with CRISPR Gold versus scrambled CRISPR Gold, which refers to a Cas9 that has a guide RNA that doesn't target MDX, or control mice, there was a 2-fold increase in the hanging time. It's still significantly lower than the wild-type mice, but it is a trend in the right direction. Based on these studies, the graduate student that led this project formed a company called GenEdit. His name is Kunwoo Lee. He's now the CEO of GenEdit. And our lab has been collaborating with GenEdit for the past few years. And on the next few slides, I will talk about a collaboration between our lab, GenEdit, and another professor named Hae-Young Lee, who is a professor at the University of Texas in San Antonio. So there are numerous organs in which gene editing can have potentially therapeutic effects. Of these, the brain is perhaps one of the most exciting. And this is because neurological diseases such as Alzheimer's and Huntington's affect millions of people every year. And there are also very few treatments for most neurological diseases. In general, developing therapeutics for neurological diseases is very challenging. And this is because of the blood-brain barrier. And also because if you just globally inhibit neuronal signaling pathways, that generally leads to very high levels of toxicity. Localized gene editing done in the brain after an intracranial injection of CRISPR enzymes has, we think, tremendous therapeutic potential because it will overcome the problems with the blood-brain barrier. It will also only edit the regions of the tissue where you do the injection. And because gene editing is permanent, you only need to do this type of injection once. And there are already numerous medical conditions where intracranial injections are routinely done. So intracranial injections in general are fairly safe. However, despite its potential, very little is known about brain gene editing. So to determine if non-viral gene editing in the brain was possible, we made CRISPR Gold and sent it to Hae-Young's lab, and she investigated if CRISPR Gold could do gene editing in the brain. And for this project, the CRISPR Gold is designed just to knock out genes, not to do HDR. For these experiments, Hae-Young used the Ai9 mouse model. The Ai9 mouse model is shown here. It's composed of a mouse that has the TD tomato gene upstream of a stop codon, so it doesn't actually make the TD tomato gene. But if you cut out the stop codon by making 2 cuts with CRISPR, then the stop codon is removed, and the cells that underwent the gene edit will now start making the TD tomato gene, which is the same thing as the red fluorescent protein. That contained Cas9 that was designed to do this, and also CPF1 that was designed to do this. We shipped it to her lab, and she injected it into the hippocampus and striatum via an intracranial injection, and then looked for red cells. The results are shown here on this slide. Every cell that's red is a cell that got 2 cuts and is edited. So you can see on the top is the Cas9 injected striatum, and under the header that says injected, you can see a lot of red cells in the yellow box. Each one of those cells is a cell that got 2 cuts, and then not only 2 cuts, but 2 cuts and recombined. And then in the bottom is the CPF1 treated mice, and again in the yellow box you see a lot of red cells. We were able to get the percentages of cells that were edited by doing a simultaneous DAPI stain. It turned out to be between 10 to 15%, and it's worth mentioning that that's actually a very high number for Cas9 or CPF1 in the Ai9 mouse model, because each cell has to not only get 2 cuts, but it also has to recombine properly, and it's the recombination that's fairly inefficient. This is actually fairly encouraging, and the conclusion is that CRISPR Gold can do gene editing in brain tissue. So based on this, we investigated if CRISPR Gold could specifically do gene editing in neurons. And to do this experiment, Hae-Young used the transgenic mouse model where YFP is only expressed in the neurons. And we made CRISPR Gold that was designed to knock out this YFP, and she then injected it into the hippocampus and then counted for YFP negative cells. The results are shown on the next slide here. The y-axis is the percent YFP cells. And again, we use 2 different flavors of gene editing enzymes, Cas9 and CPF1. So on the top, you can see the Cas9 injected mice. We do a simultaneous DAPI stain to get the percentage of cells that are YFP and also that decrease. So on the top bar graph, you can see that in control mice, 30% of the cells, about 30% of the cells on a histology slide in the hippocampus are YFP positive, and that decreases to about 10 to 15% with CRISPR Gold. And on the bottom slide, it again shows a similar phenomena that CPF1 is about 35 to 40% YFP positive, and that goes down to about 10% after gene editing with CRISPR Gold that contains CPF1. So the conclusion from this is that Cas9 and CPF1 based CRISPR Gold are very efficient at knocking out genes in the brain. And this is because Cas9 and CPF1 are really good at knocking stuff out. They're not so good at doing recombination. And if you want to just knock something out, they're reaching 50%, which is a fairly high number in the vicinity of the injection. So based on this, we investigated if CRISPR Gold could have therapeutic effects by doing gene editing in the brain. We selected Fragile X-related autism as an initial test bed for therapeutic brain gene editing. The reason we selected this disease is because of the large unmet medical need. Fragile X is the leading cause or the most prevalent cause of hereditary autism in males. It's a disease caused by a triplet repeat expansion in the FMR1 gene that has the effect of basically knocking out the FMR1 gene. The FMR1 protein is a protein that's involved in controlling the level of protein production. So FMR1 is a mRNA binding protein that controls the rate at which proteins are translated in cells. And it has a fairly, another thing I want to mention is that in patients with Fragile X-related autism, or Fragile X-related autism, their mGluR5 signaling pathway is also overactivated. So the mGluR5 signaling pathway is shown on this slide here. mGluR5 is an ion channel, and when it binds glutamate, it gets excited, it gets activated, and that leads to the production of a wide variety of second messengers. And the net result of activating the mGluR5 signaling pathway is that several new proteins get expressed, and it's these new proteins that mediate the signaling effects of mGluR5. So on the next slide, the interrelation between mGluR5 signaling and the FMR1 protein is discussed. So in normal healthy people, when mGluR5 gets activated, a lot of proteins get produced, a lot of new messenger RNA gets produced, and the FMR1 protein will then bind those messenger RNAs and prevent them from getting translated. So in normal cells, then the FMR1 protein is a negative inhibitor of mGluR5 signaling because it prevents the proteins that would normally be produced from mGluR5 signaling from actually getting translated. So mGluR5 represents the brakes on, so the FMR1 protein represents the brakes on mGluR5 signaling. However, in patients with Fragile X-related autism, when the mGluR5 ion channel gets activated, the proteins that are being produced from mGluR5 signaling are produced uninhibited, and this is because there's no FMR1 protein in these cells. So when mGluR5 gets activated, a whole bunch of messenger RNA gets made, and those messenger RNA then get converted into proteins without any inhibition. So there's no brakes on the signaling pathway, and that leads to a variety of detrimental effects and has very profound effects on learning and memory. And this is considered to be one of the primary causes why Fragile X-related autism leads to mental retardation. Turns out that the mGluR5 signaling pathway is involved in numerous neurological diseases, everything ranging from Parkinson's to drug addiction, and also Fragile X-related autism. So there's therefore great interest in being able to develop strategies to inhibit mGluR5 signaling. Turns out this is very challenging to do with small molecules because of the blood-brain barrier, and also because of the toxic effects of just globally inhibiting mGluR5 signaling in the brain. However, CRISPR-based therapeutics have the potential to inhibit mGluR5 signaling by simply knocking out the gene in localized regions, and we investigated if doing this in the striatum could have therapeutic effects. So we made CRISPR Gold that was designed to knock out the mGluR5 gene and sent it to Hae-Young's lab, and she injected it into the striatum and then looked for mGluR5 negative cells. The results are shown on the next slide. So the y-axis here is the percent mGluR5 positive cells, and this was done by immunohistochemistry, where you look for a 50% reduction in staining and count that as a negative cell. So in control wild-type cells, about 65% of the cells are mGluR5 positive. If you treat them with CRISPR Gold that has a guide RNA that targets mGluR5, you can reduce that by almost 50%. And we saw a similar result in knockout mice. So in the control FMR1 knockout mice, those are untreated. Again, it's around 60 to 65% are mGluR5 positive, and that goes down to about 30% if you inject them with CRISPR Gold that's designed to knock out mGluR5. So based on these results, we investigated if knocking out mGluR5 could rescue FMR1 knockout mice from the repetitive behaviors that are characteristic of autism. There are numerous repetitive behaviors that the FMR1 knockout mice exhibit. One of them is that they jump up and down obsessively. So that's shown on the graph on my left. So the y-axis is the number of jumps in a cage. And what you can see here is that the control mice, so these are healthy mice, have almost no jumping phenotype. But the control FMR1 knockout mice do have a jumping phenotype. So they jump about 20 times more than a wild-type mouse. However, if you partially knock out mGluR5 in the striatum with CRISPR Gold, then you can rescue this jumping phenotype. So now they jump maybe 5 times higher than the control mice, and their jumping behavior has been significantly diminished by this partial mGluR5 knockout. There are also numerous phenotypes that we had no effect on, and this is because we only did the editing in the striatum. So line crossing is another phenotype that's exhibited by these mice, but that's a hippocampal mediated phenotype, and we had no effect on that because we only edited the striatum. Okay, in the remaining few minutes, I want to talk about another strategy for delivering gene editing enzymes, and this is based on making Cas9 protein conjugates. So the first two strategies I talked about were based upon nanoparticles. Nanoparticles can be very effective, but they have one limitation, and that is because of their large size. So it's unclear if nanoparticles will be able to diffuse long distances to edit large volumes of tissue, which will be needed to ultimately translate any of these technologies. The Cas9 protein, however, is significantly smaller than a nanoparticle. It's only about 15 nanometers in size, and it should be small enough to be able to diffuse into regions of tissue where nanoparticles cannot. So we're therefore very interested in being able to develop delivery vectors that are just composed of the Cas9 protein conjugated to various polymers and peptides. However, if you just start randomly conjugating things to Cas9, you will most likely modify lysine residues that are essential for its activity and inactivate it. So to get around this problem, we made a new linker called the DEC linker shown on this slide here, which is a disulfide self-immolative linker. And the DEC linker allows you to reversibly modify proteins via their lysine residues. So the DEC linker is composed of two self-immolative linkers connected in series. It's got a thioethyl carbonate and a 1,6 elimination linker. And once the disulfide there gets reduced, the thioethyl carbonate cyclizes, generates a phenol, which then undergoes a 1,6 elimination linker, a 1,6 elimination reaction, generating the unmodified protein. So there are numerous applications of the DEC linker. I'm going to talk about two of them today. The first one is to conjugate PEG to the Cas9 to enhance its tissue diffusion in the brain. And this is under the heading of protein pegylation. The next application will be to conjugate cell-penetrating peptides to Cas9 to make a self-delivering Cas9. So in the first project, we used the DEC linker to conjugate the polymer polyethylene glycol, or PEG, to Cas9 via modification of the lysine residues to get what we're calling Cas9 linker PEG. And the idea is that the PEG will enhance the diffusion of Cas9 in brain tissue. Once this Cas9 PEG gets inside of a cell, the GSH inside of the cell will then reduce the DEC linker and remove the PEG and then generate unmodified Cas9, which can then be active and do its gene editing. And I should mention that this was a collaboration between my lab, Ross Wilson's laboratory at the Innovative Genomics Institute, and also Chris Benkowitz, who is a professor at Ohio State University. So my lab and Ross's lab made the Cas9 PEG conjugates, and then we shipped them to Chris's lab, and he investigated if conjugating PEG to Cas9 enhanced its diffusion through brain tissue. So the experiment he did was he took Cas9 PEG, conjugated via the DEC linker, injected it into the right striatum via convective enhanced delivery, and then he injected native Cas9 into the left hemisphere. And then after 1 hour, he sacrifices the mice and stains them by immunohistochemistry. And this Cas9 was also fluorescently labeled, so he also analyzes them by fluorescence. So on the right is the immunohistochemistry results. So you can see that if you just inject Cas9 by itself, it doesn't diffuse particularly well. So the brown staining here represents the Cas9. So you can see the one on the left, there's not a lot of brown staining. However, on the right, the hemisphere that was injected with Cas9 DEC PEG, that did diffuse throughout the brain tissue, almost entirely throughout the striatum, and demonstrates that reversible conjugation with PEG seems to be able to enhance the tissue diffusion of Cas9 in the brain. In addition, we've used the DEC linker to conjugate cell-penetrating peptides for Cas9. So in this project, we made this molecule called CPP-DEC, which is the cell-penetrating peptide arginine 9 conjugated to the DEC linker with a nitrophenyl handle, so that when you mix it with a protein, the protein reacts with it via its lysine residues. So we made this and conjugated it to Cas9, and then added it to cells, and then investigated if we could perform gene editing. Because of time, I'll just skip to. I'll just skip to those results. So the results of this are shown here. The y-axis is the editing efficiency, and the x-axis is the various groups. So this was done on GFP hex cells. So you can see that the red bar is the cells that were treated with Cas9 CPP conjugated by the DEC linker. We saw 5.1% editing, and if you just use the Cas9 RNP in itself, it's only 0.3%. So we were able to enhance the delivery of Cas9 in the cells using this. So in conclusion, I've talked about two delivery technologies for gene editing enzymes. The first one was Crispr Gold, which could do gene editing in the muscle and brain tissue. And then I ended with a new linker called the DEC linker, which allows you to reversibly modify proteins. I want to thank the people that did the work. All of the Crispr Gold work was done by one very talented graduate student named Kunwoo Lee, who's now the CEO of GenEdit, and all of the DEC linker work was done by Mao He, who's shown here on the slide all the way at the top. Finally, I want to thank you for your attention, and if there are any questions, I'd be happy to answer them.
Thank you so much, Niren. This is really great work, great presentation. As we all know, that the delivery of CRISPR RNP into the target organ in vivo has been the most challenging, and your lab's research really helps drive the application of CRISPR technology in gene therapy. So thank you for your talk. We have a couple of questions for you. I know that we only have about 10 minutes, so let's get right into it. Question from Brian, for the Dec Linker PEG or CPP for Cas9 delivery, would it be possible to also deliver sgRNA along with the Cas9 protein?
Yes, yeah, that's a great question, and the answer is yes. That's pretty straightforward to do. And in fact, I think we've already done that. So what you have to do is you have to first formulate the Cas9 RNP. So first complex the Cas9 protein with the guide RNA, and then react it with the Dec linker and all those things. So yeah, so definitely possible to deliver fully functional RNP into cells using the Dec linker.
Thank you. This is great work, says Kristen. Have you tried CRISPR Gold at tissues other than muscle and brain? What would be the barriers to apply CRISPR Gold at other organs?
Yeah. So that's a very interesting question. The big challenge is the tissue permeability. So that's why we're sort of hesitating on using it for other organs. So we think it's going to work really good if we're places you can do a direct tissue injection. But outside of that, it's too big to be permeable from the blood into the tissue. If I were to guess, I think it has a reasonable chance at working in tumors.
Okay, thank you for that. Is multiplexing of sgRNA possible with CRISPR Gold?
Yes, I think that should be possible. I think each CRISPR Gold has hundreds of Cas9 RNPs. Now the problem though is that if you look at our animal data, we're reducing about 50% of a single gene in the brain tissue. Now, if you had multiple guide RNAs, unfortunately the efficacy would get diluted. So I think at our current state of efficiency, we're not really efficient enough to deliver multiple guide RNAs, because we would just be inhibiting several genes at 10 to 20%, which probably may not have therapeutic levels. But I think if we could increase the efficiency, then I think we could do multiple genes.
Jen has a question regarding CRISPR Gold. How long are the gold complexes active in the cells? How often do you need to re-inject the complexes, or would 1 or 2 injections be sufficient to treat muscle dystrophin?
Yeah, so the really interesting thing about gene editing is that it's permanent. So we would only have to give 1 injection. So once you do the editing, it's basically done. Now the issue is that the big issue right now is the tissue diffusion. What we showed you was mouse data. A human muscle is going to be 1,000 times bigger. So the question is, after a single injection, can we get that diffusion? That's kind of the reason why we're so heavily focused on the PEGs and things like that. But it certainly should be possible to do multiple injections. The only issue there is the immune response to the Cas9 protein, but this is a pretty heavily pegylated and heavily encapsulated thing, so it should survive. It should be possible several injections as long as you do it within a 2-week time span.
Great, thank you. From Steve, how many gRNA and Cas9 donors can you pack into one CRISPR Gold? Is there a size limit to the donor template?
Yeah, that's not really. I mean, I think the donor template can be very large. The hard part with the donor template is that it's making it. The reason we went with the 180 base that's really easy to purchase. We just purchase it from IDT, so that allows us to do a lot of testing in terms of various formulations, things like that. We have, at one point, we did try some experiments with like knocking in large functional genes. The problem is just making that is so hard. It got so expensive, it just was prohibitive. But if somebody had a really clean source of especially single-stranded donor DNA that was long, 2 kilobases, 3 kilobases, then that would really be quite good.
Another question from Dave. Have you tested the cellular immune response to the CRISPR Gold nanoparticles? Any toxicity? What is the half-life of these particles, and how can the body remove those nanoparticles after Cas9 localization to the cells?
Yeah, great. So the gold is not degradable. So the gold will sort of hang out in the cells for a long time. Everything else is basically a biomolecule. It's either a peptide based or a nucleic acid based. All that's going to hydrolyze in individual amino acids or individual nucleotides. The gold nanoparticle will persist, but gold, people have done studies with gold. It seems to be fairly inert, and cells and tissues seem to tolerate it. So that's the one limitation of CRISPR Gold is the gold. We are trying to actually make new formulations that don't have the gold, and I think we've succeeded at doing that. The immune response, we did measure it actually in that paper. We did do a big cytokine analysis in muscle tissue. So if you do CRISPR Gold injections and then look for cytokine levels in the blood, or even immunoreactivity or macrophage presence in the muscle tissue, we didn't really see any major increases.
And one last question from Victoria, who says, thank you for the talk. For the Duchenne project, why do you think the muscle strength for gold CRISPR sample did not improve much in comparison with the WT?
Yeah, right, great. So I mean, I think it's because we're only seeing about 5% HDR, so that's sort of, I think it's the bare minimum to where you would start to see clinical benefits. And I think also we're really seeing HDR in the local vicinity where we're getting the injection. So this, I think the CRISPR, because HDR is such a complicated process, we're doing injection. I think we're editing, I think 3 to 5 millimeters in the vicinity of the injection. And even if we do a lot of injections, it's still somewhat limited. So I think that's the big one. It's just the amount of editing we're seeing. We need to increase that, and once we do that, I think we'll see better muscle results.
Thank you, Niren. Once again, many thanks for the presentation. We'll let you go, and we hope to see you at the next one. Much appreciated.
Thank you very much. Yeah, it was great. Thank you.
Thank you. Bye.